Hi, everyone, and it's my privilege to be moderating this session, which is focused on addressing the global cancer burden through early cancer detection, but also strategies that aim at reducing inequities in access to appropriate care and therapies. So I'd like to start by having the panelists introduce themselves, followed by a brief synopsis of where they see the greatest challenges and opportunities when it comes to advancing early cancer detection, but also ensuring appropriate access to cancer care and new therapies. Victoria?
Okay, thank you so much. Thank you for this opportunity to be here. So I'm Victoria Zazulina. I represent a healthcare business of Merck, KGaA, or EMD Serono as it's known in the United States. So I represent clinical development, and for me, if you think about how do we address the global cancer burden, there's multiple elements how we can do it, but what I'm most passionate about is thinking that it's always better to address the disease at the earlier stages, and no wonder we're all chasing screening programs, early detection programs, etc. But what is important for us when we are discussing the possibility for those screenings and early detections is that it needs to be paralleled and matched by the opportunities to act on it, because there's little value in detecting something but then saying to a patient, I'm sorry, this doesn't look bad, but maybe I don't have the best option for you. And that's where I see the opportunity for us and a call to action for us as pharmaceutical companies and drug developers to bring more drugs into earlier disease settings so we can match the opportunity to detect, but also to quickly act on it. And another aspect that you've already touched upon that, Sean, is that this equity, the disparity between healthcare access and the systems, not only within the US, there are some disadvantaged populations, but also globally, we see a lot. The majority of cancer deaths actually are happening in the low and low-middle income countries, up to 70% of all the cancer deaths annually. And I'm sure even in the United States, if you look at the parameters, if you look what constitutes those numbers of deaths, the majority probably will come from the communities which are underserved. So the question for me is how do we stretch our data, the use of our drugs, and generation of real evidence that is going to be convincing to bring those drugs to those communities and encourage them to participate in our clinical trials.
Okay, thank you. Craig?
Yes, good morning, everyone. My name is Craig Tendler. I head up Oncology, Clinical Development, and Global Medical Affairs and Diagnostics for Janssen, or Janssen as we call it in Europe, Research and Development. And I also want to thank Sean and the meeting organizers for inviting me to participate in this panel. When we think about the disease burden, it is with a lens of trying to reduce the risk of developing metastatic disease, because that's the setting where, as we've heard in this conference and elsewhere, the cost to society is greatest, the likelihood of cure is really reduced. And so it's, for us, we have a focus on intercepting what we call disease interception, which is really about trying to identify patients at highest risk for developing metastatic disease and using some of the drugs that have early evidence of activity in the very resistant refractory setting, but bringing them earlier into the setting where you can have a potential for cure with fixed duration of therapy. So, for example, in adjuvant therapies for high-risk patients. And that requires a few things, right? That requires an ability to detect that population at an earlier time point, to understand what constitutes higher risk for that patient population, to be able to provide the access of the intervention, especially as it starts with a clinical trial, and also to have earlier intermediate endpoints that are likely to predict the clinical benefit while you're waiting for the long-term outcomes to mature, which I think is also something very much in line with how the FDA is thinking about this as well, to really encourage pharma sponsors to pursue these type of development programs in earlier stage disease, something that they call now Project Frontrunner. So I think as we discussed today, there has to be a focus moving away from that single-arm phase 2 trial where, a company, a biotech, or a pharma sponsor is putting all their efforts just in that first approval in the most heavily pretreated resistant refractory population. We have to start moving away from that as sort of the strategic goal and start thinking earlier and earlier how we're going to bring our therapies into the patients who are at highest risk for relapse, where we can still have a reasonable chance of cure.
Thank you, Craig. Tracy?
Good morning. Thank you so much for being here and for inviting me. My name is Tracy Baroni Allmon. I'm the Vice President of Global Oncology Access and Reimbursement Policy for Takeda. I think that once the very real, well-defined needs already presented by Craig and Victoria are met, we have a real need to recognize what are the limitations on access. It's hard to do that globally. But if I could name 3 things, because I think 3 is a good number to kind of try to remember and work around, value assessments are very important, and yet they differ so much from region to region. They're not required everywhere, obviously not in the U.S., and yet we see a movement toward that, not just with ICER, U.S. ICER, but also with the IRA and how they're going to think about setting a price for a drug after it's been on the market. The second I would say needs to be addressed is silo thinking. Everybody talks about the costs of drugs. Nobody wants to talk about the benefits and the money that they save, the child care that's maybe not needed because somebody can take an oral and not be seen every week or every other week for chemotherapy and be down for 3 or 4 days recovering. They don't think about the burden to the workforce, the burden to the caregivers. And so I guess it's not that they don't think about it, but there's not a good way to quantify it. And even HEOR data is not sufficient. And then I think the third aspect that I would think about is the need to educate, incentivize, reward, and maybe even penalize at least some of the stakeholders who aren't doing what they need to do to ensure that the cancer burden is relieved. So is it physician education? Is it payers recognizing that they shouldn't be the last say and there need to be discussions? Is it patients and the need to educate them, not just on the system, but on cancer, on next-gen sequencing testing, on all the things that are going to be important to them and their family as they go through this journey? So those are my 3 from an access and reimbursement perspective.
Tracy, since you mentioned IRA, or the Inflation Reduction Act, which, as many of you know, was signed into law by the President in August of this year, empowering the HHS Secretary to negotiate drug pricing. How do you think, Tracy, this act, once these provisions are enforced in a few years, impacts the development and delivery of new medicines, anti-cancer therapies in this case, in the United States, and how does that compare to some of the strategies that have been employed outside the United States by federal authorities?
Thanks for the question, Sean. I wish I had a crystal ball. I do not, but I think you have to take into consideration the cost of innovation, the cost of failures, which is something that nobody at HHS wants to talk about, right? The drugs that are, the programs that are cut, left on the lab floor. I think that there may be companies, and companies may have to, make different decisions when you look at a patent life of effectively 9 years for a small molecule until you get price controlled, or 13 years for a biologic. We've already seen 2 companies within the last probably 10 days give blame to the IRA for decisions that they made around development. I think that because this is only for the Medicare population, maybe drug development goes toward non-Medicare populations. And that's certainly not anything that we want to see. But when you take a big sector of the oncology patient population and squish them into a price-controlled setting, it could ultimately disadvantage Medicare patients.
That makes sense. Victoria, you mentioned that in your work, you're always thinking about how to stretch the data that you're generating to address some of the issues that we've been talking about, especially in the context of addressing the needs of historically underrepresented populations. And, you know, with what we're starting to see in terms of the downward pressure on drug pricing and some of the uncertainties that are involved with the evidentiary standards that are required to power some of these negotiations in the U.S., but also the evolving needs of ex-U.S. Authorities to go beyond just regulatory approval of a therapy in order to accommodate access to it. So how are you thinking about that in the context of clinical development and generating the evidence that can carry forward into the post-market setting and the post-approval setting? As an example, we heard a lot in the past couple of days about extending the reach of clinical investigations to underserved communities through decentralizing clinical trials. And clearly, some of that evidence in those populations are needed in order to provide access post-approval. Is that something that is being entertained and paid attention to in the context of the evidence package during clinical development?
Yeah, very much so. I think there's a few questions, I think, in what you just raised, Sean. So if we talk about the underrepresented population, this is becoming a very big topic for our clinical development programs. The issue is that with early stage drug development in oncology, and we're still not doing completely safe therapies, right? So we have to control, especially first stages, phase 1 development. You might be prepared to see some surprises on safety, even though you kind of de-risked it from your preclinical development, you think you can predict. So going fully decentralized into underrepresented tertiary community centers and doing something there with very early stages of drug development might not be an opportunity for each and every drug. Let's be honest about that. But as the drug package matures, yes, absolutely. As the more we learn about it, we're now encouraged more to go out there to create certain algorithms or look for the real-world evidence, or we are looking for the companies that can provide us with test results, because, on Tempus or any other foundation medicine, right? So those who are working with the totality of the market, they could tell you there's a hotspot here, hotspot there, and that's where you drive your clinical development effort. That collaboration is enhancing and getting very much stronger to find the right patients and then ultimately decentralize or reduce the burden and complexity of our trials, because sometimes it doesn't have to be fully decentralized and we might not have the opportunity to do so, but definitely making our protocols leaner. And that a little bit goes against that, you want more data, you want more evidence, right, to be more convincing. But then you're becoming prohibitive in terms of what patients can take, what physicians can take, healthcare professionals who are looking after them. So this is always that trade-off that we are discussing. But ultimately the direction is that we need to find those patients, stretch out. We're looking at larger cancer centers to try and tell us with the satellite sites if there could be referrals or if we could use the network of this hospital to somehow better identify the patients, to bring the drugs, to bring the clinical trials to where the patients are. So that was one question. I think that was one part of the question. Another one you asked about the real-world evidence and what are we doing to enhance the package and make it more representative. So here, there have been, if you allow me to say, some hit and miss, messages also from FDA and how they behaved in terms of looking at the real-world evidence. We know about some successes, for example, registering CDK4/6 inhibitor in male breast cancer. That was great. But fairly recently, for example, there was an ODAC where FDA, or the advisors for FDA, voted against the real-world evidence provided as a control arm to a single-arm data for a drug with high unmet need, because they said it's not contemporaneous, it doesn't meet certain requirements. So, I think the key ingredient here is that really being aligned with FDA and upfront negotiating and understanding each other much, much better, because it's new for both sides. Nobody knows what the gold standard is, so we're arriving there together. So I think the key is that dialogue, openness and the dialogue upfront.
Sure, that makes sense. Speaking of gold standards and how in oncology, a lot of times we actually don't know what the gold standards are, including some of the endpoints that we use, and thinking about how to validate these endpoints. Craig, in terms of disease interception and cancer interception, which is a fascinating topic, but it does rely on intermediate endpoints, as you touched on. And now with the advent of liquid biopsies, that can give us a lot of interesting information at a much earlier stage of disease, even before any signs and symptoms of disease are apparent. How do you think about defining these intermediate endpoints earlier in the course of disease and actually clinically validating them in a manner that would be able to support development programs?
I think that's an area where all companies are looking very keenly to beef up their research efforts to provide that evidence package. Fortunately, in many tumor types, we do have markers that are easy to detect now, whether it be detection of minimal residual disease in hematologic malignancies or circulating tumor DNA, which can be done with high sensitivity, reliability, reproducibility. And what we try and other sponsors try to do is collect, in collaboration, often with across companies, collect that data and share it with the FDA and get to a point where at least the endpoint, the intermediate endpoint, can be considered likely to predict clinical benefit. It doesn't have to be, and Dr. Pazzer has been very clear about this, it doesn't have to be a true surrogate where it's established as clinical benefit. It has to have enough evidence so that when the long-term outcomes mature, that there's a reasonable chance that they will correlate with the change in the intermediate endpoint. And I think for most of these settings, we have that kind of data and in fact are designing trials such that there is an early look based upon a change, the proportion of patients who achieve that clearance of ctDNA or achieve undetectable MRD. But the study is powered for the long-term outcomes. So we have confidence as the sponsor, health authorities have confidence that within the same trial, we will be able to determine if that early endpoint actually delivers that benefit in terms of improvement in progression-free survival, overall survival. And again, I think that's very much in line with this new Project Frontrunner that FDA has put out through the Oncology Center of Excellence, because it is giving the opportunity for sponsors to think about designing trials with a intermediate endpoint where you could file based upon an early look and still provide therapy upon approval without having, while continuing to conduct the trial, but not having to wait 7, 8, 9 years until you find out that the drug is actually delivering substantial clinical benefit. And so to me, this sort of balances the original accelerated approval strategy with the need for sponsors to design trials that appropriately determine clinical benefit, not just for health authority approvals, but also for the impact on society and for ensuring the adequate evidence for reimbursement.
Sure. Makes perfect sense. I'm glad you highlighted the concept of surrogacy in terms of the endpoints that we use in oncology and intermediate endpoints. It does turn out that a lot of the endpoints that we do use for drug approvals, like overall response rate and progression-free survival, their surrogacy from a technical mathematical perspective has never been established. In fact, these endpoints were generated based on consensus in the community that these are endpoints that are important to patients. When a tumor shrinks, it is an important event, and regulatory authorities align themselves with that consensus. So these surrogate markers were, in fact, never mathematically established. So as we think about new intermediate endpoints, for example, those emerging from liquid biopsies, who should establish the clinical utility of these endpoints? It's hard to imagine that every company will have a unique, singular approach to validating the clinical utility of an intermediate endpoint that's bespoke to their development program. So how can we generate consensus around these intermediate endpoints, perhaps in the context of pre-competitive collaborations, to generate that consensus? Victoria?
I think you've answered your question already. In the pre-competitive space, I think all of us need to work on that because these are big data sets which, to establish this surrogacy is doable, but you need to have so much data, right, in a statistical fashion to confirm that one thing predicts for another. That within one company, within one drug, you might never reach that number. You might or you might not, right? And I know many companies are embarking on this journey in the interest of a particular drug, particular indication, but then every indication is different. What works for, I think in lung cancer, PFS, OS have been reasonably well linked, while in other indications they're not. Also different drugs. We know that PFS and OS for immune checkpoint blockade is not necessarily, PFS is not as remarkable, but OS is actually great, right? So that it depends on the drug. And I think going back to this pre-competitive space, that's where we could all collaborate, large institutions. The data set from MD Anderson, looking at what they're measuring, regardless, we could take disease and zoom out of the interventions that have been made, for example, which drugs have been used, but looking at ctDNA, and I know they're very keen on MRD, on the adjuvant setting, and looking at what happens there and how long the patients live, regardless of which intervention it was. If they cleared their DNA, what happened? Did they live longer? That already helps to establish. And then, of course, pharma with our data sets, I think we just need to formulate key questions because there's so much you can do, but you have to start somewhere with one or two, maybe classes of therapies or maybe indications which are at the highest sort of interest. And I think it's a call to action.
Mm-hmm. Sure. And it seems like at the heart of it is data and ensuring that we advance data liquidity. Obviously, efforts like Project DataSphere have certainly moved the needle in that direction, and hopefully now in the context of pre-competitive collaboration, especially with these intermediate endpoints that are necessary at this point to move earlier in the disease setting, hopefully we'll see more data sharing. And speaking of intermediate endpoints and clinical utility, the approach that a lot of next-generation diagnostic companies are taking right now is once their assays are analytically validated, and this is a common motif, especially in the realm of liquid biopsies, they generate the clinical utility, the data necessary to show a clinical utility in their negotiations with payers, meaning the FDA, currently at least, does not preside over monitoring and regulating laboratory-developed tests. So the clinical utility assessment and the data generated for establishing clinical utility is part of the payer negotiations. So Tracy, how do you see that evolving over the next few years, or in terms of the existing dialogue that these next-generation diagnostic companies are having with payers?
It's a big question, right? Because I think that the three items that I started with, that touches on every one of them. You have to have a health infrastructure within your country, region, that identifies innovation and rewards innovation and ensures that there is a path for that innovation to get to patients. Nobody's innovating so that we can talk about it. We're innovating so that we can get it to patients and then learn from it. So I think that when you think about budgets, healthcare budgets, program budgets, country budgets that pay for healthcare, they need to recognize, there needs to be a general recognition that access to the tests, whether it's through a public-private partnership, whether it's through a freestanding foundation, whether it's through the normal payer cycle, is imperative. And we need to think about how do we work with policymakers to get those public-private partnerships started, to get the access, to create the track record, to use the data, to then show that this should be standard of care in every country. When you have a targeted therapy or the possibility of identifying a mutation that will allow a patient to enroll in a clinical trial, access to that is imperative. We, as the industry, aren't usually allowed to pay for that, nor is the testing company. We've got to find a way to fund that access and then learn from it across both the both the commercial access space, but then also the pre-commercial.
Tracy, a quick follow-up question on that. Do you think payers in the U.S., but also outside the United States, are close in their thinking to what you're mentioning in terms of these diagnostic assays, really accommodating access to these diagnostic assays at the point of care? Do they know what they should be looking for in order to accommodate access at the point of care?
I was fairly surprised by Dr. Mitchell's comment earlier around lack of understanding of testing and the importance in lung cancer. So I have to say that being a new piece of information for me, I'm still going to say the U.S. is in a much better place than the rest of the world from an access to testing perspective, a recognition of what it can bring to the patient and to the overall scope of the treatment. I do think, however, that there are still challenges, right? Can you get a second test if you're a Medicare patient, whether it's because they think the same cancer has further mutated, or whether it's because 2 years ago you had a cancer diagnosis and got tested, and now you need another test. And so that's a challenge. Also, I think because there are so many payers in the U.S., providers aren't always sure if the test is going to be covered, and so sometimes they shy away from giving the test, or they scare the patient away from it. And there are stories where patients are told, this could cost you $1,000 or $2,000, and this is just the beginning of your cancer journey, so let's skip it for now. That's scary. So I think there are a lot of things in the U.S. that are going well, but there's room for improvement.
Since we're talking about early detection and prevention, including primary prevention, which has been an aspiration for many in oncology, thinking about cancer vaccines, for example. The recent advent of mRNA technologies that have really effectively reduced the burden of disease associated with SARS-CoV-2 infection is now moving into the world of oncology. For example, as evident by the recent collaboration between Merck and Moderna in the setting of patients with high-risk melanoma. So Craig, as you think about cancer interception, disease interception, how do you think cancer vaccines play a role in that equation?
So, it's always interesting to sort of look back on our training and how things are now completely antiquated. So I started in drug development in 1995, actually first collaboration with John Kirkwood, who was developing vaccines for melanoma. And at the time, it was just very difficult with those type of peptide vaccines to get a specific anti-tumor immune response. And it's interesting how, the field kind of shut down and now, of course, reinvigorated with better knowledge of how to activate T cells and how to create memory cells and responses, and how to build synergistic, combination regimens. And I think that, to me is the most interesting aspect, to be able to think about putting different types of immune oncology agents together where, you're addressing the deficits of one, let's say, PD-1s, and they obviously have, tremendous benefit, but there's a large group of patients that aren't benefiting, and how can you still utilize the knowledge to reactivate the immune system in what we call cold tumors, which don't have much of a immune activation potential, or in a melanoma setting where you're looking to, bring the benefit to a higher proportion of patients. So I think it makes a lot of sense if we can use mRNA technology, to develop COVID vaccines and turn on an immune response against a virus. We should be able, with the right antigens and neoantigens that are well known in, for example, melanoma, to be able to do that for patients. And I think the exciting thing now is that the technology is available to do that. And again, in a setting like, melanoma, to bring that to early stage, we know how to identify patients who have high-risk disease after surgery. And again, why just focus on the heavily pretreated when we know we need a highly functioning T-cell repertoire and immune system to be able to benefit from activation with a vaccine approach in combination with PD-1 directed therapy. So I think we're there. It's the right thing to study, and we have to just also pick the patient population that we believe is likely to benefit as well.
And if I may add there, actually, this mRNA technology has actually started in oncology. It was interesting how initially the first attempts of mRNA companies trying to combat cancer did not really yield much result. And then COVID happened, and now there is a great attention back to the same technology because we have proven that it's scalable, it can be produced, it can be delivered on a wide basis, etc. But the question about the mRNA vaccines is also, talking about primary, secondary prevention. You have to have a disease, in this case for mRNA then to prepare your dendritic cells, etc., to act against that disease. And there are attempts actually to do personalized mRNA vaccines already now for cancer patients. Can you take this into primary where the patients do not yet have cancer? Mm-hmm. Maybe, but that's again another field probably for pre-competitive assessment where which neoantigens could potentially be those that are expressed in this particular disease. There's a lot of AI and I guess machine learning from multiple samples of patients with melanoma, with lung cancer, and with many others that could later inform how can you construct a universal vaccine that could completely prevent the appearance of those cells. But that's a big dream.
Well, it makes perfect sense, and I think we're moving in uncharted territories where some fundamental questions are being raised. What is having cancer? What is disease? If we have an asymptomatic individual with no signs or symptoms of a malignancy, but a positive liquid biopsy result, what do we do with that patient? Does that patient actually have cancer? And do we intercept? So there are a lot of interesting fundamental questions are being raised, which actually in a way came up during the last panel discussion where our disease classification schemas have been anatomical and histopathological, not mechanistic. Now we're becoming a lot more bottom-up and looking at the mechanism of disease, which is probably the way to go, especially if we're talking about disease interception. Being told we only have a few minutes left, so I'd like to open discussion for questions. Dr. Murphy.
What a great panel, and it's good to see you on the stage, Sean. If I remember correctly, it was about 8 years ago when you graced Dr. Goodnight's SAS stage for the first time, and we all had an opportunity to see what good-looking men's hosiery is all about. You'll remember that the 41st President of the United States shared your taste in socks. And we're glad to see those socks back on you here. That's the editorial. Here's the comment. It's an ill wind that blows no good. And during these 2 days, we've made comment about some things that COVID has brought to us that really has been advantageous, that otherwise probably would not have taken place. Under emergency circumstances, decisions were made, policies were set aside, patients were not put at undue high risk, and progress was made. And billions of this world's inhabitants have been spared a scourge. Let us not go back to that pre-pandemic default. And I'm speaking now also specifically of our regulators. The one chair that's absent or open should hopefully next year be filled by the person who you were at that time 8 years ago, the founding Deputy Director of the Oncology Center of Excellence of our FDA. We need the FDA on that stage. Not having the FDA right now, however, doesn't stop me, Sean, from asking you no longer wearing an FDA hat. What from the pandemic would you want us to save with regards to regulation for all three of you, as expert indeed as you are, and as devoted and dedicated as your careers have been and brought you to this point? Everything you do, everything that we do has to pass through that orifice of regulation before it gets to the other side when drugs can be prescribed. The FDA is absolutely essential. And boy, do we ever listen when Rick Pazdur speaks. He can attract by an attractive policy, or can, I don't want to say repel, but clearly put up significant challenges with a negative one. How can the FDA help eliminate disparities in clinical trials by attraction, and maybe also that cane as well? Thanks, John. And I'm so glad to see those socks back.
Thank you, Marty. So maybe, Craig, you can make some quick comments about this. You also already mentioned Project Frontrunner, but there are a lot of lessons learned in the past couple of years related to how we start to think differently about recruitment and conducting clinical trials, and in the setting of perhaps more flexibility from regulatory authorities.
Yeah. Well, first I want to go on record as saying that, in my opinion, they're among many, many helpful organizations. The FDA really stood up and clearly gave us guidance that helped us, number one, maintain patient safety throughout the trials and throughout the pandemic, but also, very importantly, allowed us to maintain the integrity of the clinical trials. Because without that, some of these trials would not have been able to continue and come ultimately to a regulatory decision. So that was very, very helpful. But I would say that very positively, many of the things that we were forced to do, and many drug sponsors implemented several activities that I think are now commonly done today, and we're not going to go back to the old ways of doing things. So that includes, allowing patients to get their labs, their disease assessments outside of the main center that they're receiving the clinical trial treatment. Being able, we've talked about, e-consent forms, not having to have the patient in front of you, to have the right procedure and privacy and legal policies in place that enable an informed consent to be done remotely, which has implications not just during a pandemic, but also, on the topic of making our trials more accessible to underserved minorities. Also, just being able to, evaluate from a statistical point of view what happens if a patient missed an evaluation, and trying to call that out in the application so that it's not something that, is not manageable. And a lot of the guidance that the FDA gave really helped us and other drug sponsors, deal with that situation, which was inevitable during the pandemic. So I think everything is in place to really facilitate what we call decentralization of the trials. We now have to build on that, and with an eye toward making our trials more accessible to patient populations, underserved minorities who cannot always get to the main clinical trial site. Sometimes they can receive study medications, especially if they're oral. We were doing a lot of, drop shipments right to patients' homes. These are the things that we continue, and I think are, in some ways, the silver lining of having to manage through a very challenging situation like the pandemic.
Thank you. Any other questions? So with that, fortunately, we're out of time, and I'd like to thank the panelists for sharing their valuable insights. Thank you.
Thank you.